I'm from the group of Lars Kaderali from the Institute of Medical Informatics and Biometry at the University of Technology in Dresden. So unfortunately, Lars could not come today, so it's my pleasure to present this talk here. So just a short overview of what I'm presenting today. So first I give a background about systems virology, so what we are doing in our lab, and I'll explain a little bit about molecular and cellular models of viral replication, and then I try to bridge the scale, so from immune response and tissue spread, and then finally to the organ and to the virtual patient. So systems virology, viruses are normally not that complicated, so you have the viral genome and the hull proteins, and so it seems to be not too complicated. However, the host machinery has to be taken into account, so it's not just sufficient to study just the virus itself, but we have also to consider the host processes which are involved for the life cycle of the virus, and also to model the link between the molecular process with the clinical phenotypes. And so for HCV, for example, you have HCV can stay silent, a cell can be infected with HCV. However, it can take years until really the patient suffers from the infection. And what's the underlying mechanism? So how can this be silent for so long and then become very severe? So here, as I said, HCV, it's a plus-strand RNA virus, and it's the cause of hepatitis C. As I said, it can be silent for years and then chronifies, and for about in 80% of the patients, and then it becomes really severe and life-threatening. So liver cirrhosis and liver cancer can be a cause. Currently there's no vaccine against HCV, and the standard treatment is effective in only 50 to 80% of the patients, and this treatment has severe side effects. So the effects of the drug and the treatment, so coming from the infection and the replication of individual cells. So this is what we did in our group, so make models of the immune signaling in individual cells. But this is obviously is not enough. We can see whether the cell is infected, but why is the cell, why is the patient not showing severe effects or severe health problems for years? And then obviously we have to take into account the spread of the infection. So how many cells are infected? How do they infect each other? And what's the immune response in the cell population? And here in this red box, these are things which we currently doing in our group. And then the next step is, of course, the effects of the infection at the organ level. So, the latency of this infection, for example, the cirrhosis, and so on. And then from the organ to the clinical phenotype. So, what's the explanation for? How can this be that some people show severe effects and for some it's not for years? So coming to the next step, molecular and cellular models of the viral replication. So here you can see now the life cycle of the hepatitis C. So here the virus particle enters the cell, then it's uncoated, and then the plus-stranded RNA is translated at the end of this multireticulum, and using the host machinery and the proteins translated from the virus, after replication of the virus, the new viral particles are assembled and exported to the cell, and then they are released to out of the cell and can infect other cells and So very shortly, this is a model process, so the entry and the release and the assembly is not taken into account. However, already only for the translation and replication process, we have 11 ODEs, 15 parameters, so it's quite a complex model already. And here you can see some results. So where we tried, or we used our model to find parameters for high permissive and low permissive cells. And what we could see with the similar model here fitting for two different cells, high permissive and low permissive, the same parameters, and well, the only difference is the host factor. This was what we find out, that the host factor is the main thing here, which makes a difference for the high permissive cells having a shorter time period than the low permissive cells, and also here the number of molecules is smaller for the low permissive cells, and the explanation which we could found and also which we validated is that this is due to the host factor and not to the virus itself. So looking at the intracellular level, as I said, we have the host factors which are important, and they have of course to be modeled with the infection and uncoating of the virus. So here this is the life cycle of a virus. Then here the replication, this is what you have seen, which we modeled in the ODEs. And then of course also the assembly and release has to be modeled. So it's not, there are several different things which have to be taken into account, so it's not enough to just find the host factors or to just model the replication. Also, for the immune response, so here we have models for the WIC-I pathway, which triggers the JAK-STAT pathway, and then interferon is released, which leads to an antiviral state of cells, and this all has to be taken into account. However, there are several problems, of course, that's not easy. How can the host factors be integrated? So we can identify them using large-scale screening, but how can this be used for the mathematical models? So it's to combine this data here with ODEs, and then also the combination of the replication model with models of the JAK-STAT signal transduction is difficult. So we have, when we, for example, want to, so what we tried in our group is to combine different models, but then is the problem, they have been established in different cell lines, different experimental conditions. The underlying assumptions are not valid, so volume of cells, cytoplasm, and often the biology is unclear. So interferon, as I said, is the immune response, but how can these influence the viral replication? Where does this take place? And of course there are these technical problems, but where we heard this today quite often now, there are some standards, SBML, and we don't think that's the biggest problem. However, joint work is needed to solve this. And for example, we have a collaboration with Professor Knapp on integration of the drug effects. And so currently we realize this mutual visits of the students. However, of course, these software, the BioStorm might also help to make it easier to exchange ideas and models. So here you can see this is HIV now, the cellular factors involved in HIV, and as you can see, it's a big network, and a lot of bioinformatics has been used to establish this model, and how to translate now this into the model. This is obviously not easy. There are, for example, other things where you can use protein interaction networks, then you detect the functional modules, so not only which host factors are important, but where are the modules, so where in the cell is something ongoing, and then identify here, combine these the functional models with RNA hits, which are enriched here in these models. And here you can see has been done also in our group, where we combine exactly RNA hits, so the knockdown of individual genes, this protein interaction network. And then here you can see these colors are hits of different viruses. So where we can combine, differentiate which virus needs which module. And from this, subnetworks can be constructed. But however, the problem is how can such factor be integrated into mathematical models? So one approach or one solution would be you have the experiment, you have some black box magic, so you have a network inference algorithm, either machine learning or ODEs, and then you have the networks and automatic translation into quantitative models. So, but what's the model parameters? So on the molecular and cellular model level, there are already a lot of problems, and then coming to the immune response, it gets even more complicated. So as I said, here you have all these factors which have to be taken into account, and then in addition, of course, the cell population. So without looking at different cells, it doesn't make sense to look at the release of a virus when there are not other cells which can be infected, of course. So here you can see the spread of the infection and the immune response. So virus basically can infect uninfected cells, and then they get infected, and the infected cells then replicate the virus. You have new virus particles, and infected cells also secrete interferon, which then is used to bring uninfected cells into an antiviral state. And these cells cannot be infected, and however, it's not at all clear how this is going on. It's what we observed is that it's a stochastic process, and how can we model the stochastic spread of interferon and the infection in cell population. Currently the models of different scales are independent. So you have either you're using the cell population model or intracellular model. But how much detail is really necessary? So what we discussed before. So is it enough then to look at the bigger perspective and say, okay, the cell population is enough? It's not necessary that you go into the details of the individual cell. What's the cell variability? How can we address this in multi-scale models? And of course, what I said earlier, these different experimental conditions and all the different time scales. So it's not only that we measure, for example, the experiment at different time points. Some biological processes just need different time points. So the replication of a virus can be within 3 days. However, the spread in the tissue takes days to weeks. And then the chronic infection can take up to 20 years, so without having seeing really a phenotype. And this is problematic. And finally, then from the tissue to the organ and to the patient. So the step to the organ is very complicated already. So we talked about this earlier. So there's this virtual infected liver, which is a project in Germany where a lot of money is put into this project, and there are many groups involved. However, I think it's already several years running, and it's really hard to model this. So it's necessary that you have well-documented assumptions to integrate different models, there are so many different approaches. So you can have differential equation models, Boolean variables, stochastic models, qualitative models obtained from these network inferences, and to take all this together is just not doable just by one group. yeah, I said this before, however, there's the link to the clinical phenotype. I think this is really the problem. So do we need then the virtualized cell, so for the virtualized patient? So yeah, so we have very complex models for the cell already. However, is this necessary? And many factors are still unknown for the cell. So we have many these protein-protein interaction networks, and all this is given, but there's still much things which are just not known. For many viruses, it's not known how they come into the cell really. So what are the ongoing processes? And there are so many subtypes of different viruses, and it's not at all clear how this is what's going on really. Yeah, so this is basically the people which are involved in all these projects, the people from my group, so from the TU Dresden, and also from a former group of Lars in Heidelberg, the Virobrand, and here the people, the biologists from the molecular virology, and other people which also are involved in the project. And yeah, thank you for your